Yeah, I'm going to talk about nitrogen. But in the Ocean Sciences section of AGU, we'll probably think of Rachel Carson for just the things that Jim just reminded you of. The books, The Sea Around Us, but that was bracketed by the two other books in that trilogy. And The Sea Around Us was first published in 1951, and my own version was 1961. I think I bought it used many years later. But just as relevant as Jim also mentioned was the book The Silent Spring, which was about the effect of pesticides on birds and other aspects of the ecosystem. And at the time, but the main message of that book, which is very relevant also to oceanography, was the detrimental and widespread, the overriding message was the powerful and ubiquitous effect of humans and human activity on the natural world, including the ocean. But I bet that Rachel Carson and probably no one else at that time really ever thought that humans would be able to have an impact on the ocean in terms of the same way that they were in the terrestrial ecosystem and birds. But in the intervening 60 years, humans have really shown themselves to be quite powerful agents of global change and affecting the environment, and even to the extent of affecting the nitrogen cycle in the open ocean. So we're now at a time of rapid change in the environment, especially with human impacts, both direct and indirect, in many aspects of the environment. So it behooves us, I think, to figure out how the ocean works now, so in terms of being able to understand both the future and the past. And so I would like to begin by thanking the people who nominated or voted or were otherwise involved in bestowing this honor on me, and I'm greatly honored to be here. When I looked at the list of previous people, I figured Jim had made a mistake and called it the wrong person. Of course, it wasn't email, not a phone call these days. But I'm really, really honored to be in that list and grateful to be here today. So the people that I would also like to thank are the nitrogen babes. These are the fairly recent membership of the Ward Lab, shown in a beautiful fall day at Princeton University. Now, we've had several t-shirts, and you can see that they're quite flattering, and there are two styles. And I just wanted to acknowledge that Debbie Bronk was the original nitrogen babe, and she's the one who actually bestowed the name on the group. So we've been the nitrogen babes for decades. It just sounds better than nitrogen guys. You know, it's a generic term. And it's actually a really useful t-shirt because I really like the back the best. And so what we call that is the anatomically correct nitrogen cycle. And this particular t-shirt was designed by Andrew Babin, who at the time was my graduate student, and he, in the midst of doing some of the experiments, whose data I will show you today, setting up to measure the contents of thousands of excitaners on a cruise in the eastern tropical North Pacific, found a few moments of boredom to design the newest version of the Nitrogen Babes t-shirt. And it includes, or it shows, most of the important components of nitrogen, the compounds in which nitrogen occurs in the ocean. It shows you the transformations amongst them, and even the code names for some of the key genes that encode the enzymes that make those transformations happen, and some of the fractionation factors associated with those transformations that were known at the time of the t-shirt. So I'm going to use this to guide you through my talk on the nitrogen cycle, or the pieces of it that I've been interested in of late. So when we think about the reason that nitrogen is important, and I think that Rachel Carson would have been a nitrogen babe and would have condoned our interest in nitrogen, because it's one of those compounds like that Paracelsus talks about. A little is good, more is better, but a lot is generally bad. And so too much or too little nitrogen is of relevance in the ocean. You have too much, you have too little, because nitrogen often limits productivity in the ocean. I'll talk a little bit more about that. And too much, because too much leads to eutrophication and oxygen depletion and other downstream effects. And when you talk about too much or too little nitrogen in the ocean, you're generally talking about fixed nitrogen. So that's everything in the shaded Pac-Man shape there. And any nitrogen compound that has an NN bond is really hard for organisms to access. And so that's the non-fixed nitrogen. And so the total inventory of fixed nitrogen in the ocean is all of these components, which are considered to be biological or biologically available. So what I'm going to talk about is the processes that set that inventory by producing and consuming fixed nitrogen. Can we turn down the lights so my pictures are a little bit higher? contrast? Anybody know how to do that? No? Okay.
Okay.
Yeah, well, anyway. We have a big timer. Sorry, never mind. It's not, they're all going to be a little faint. Sorry about that. So this is brilliant pink, outlining the arrows that influence the total inventory of fixed nitrogen, from nitrogen fixation going into the rest of the cycle, and then the processes that lead to loss of fixed nitrogen. And so those are the ones that I'm going to focus on today, and you'll get to know those a lot better. And I'm also going to talk about nitrous oxide because there are lots of reasons to be interested in nitrous oxide, not just because it's involved in nitrogen loss. There's a sort of assumption that the nitrogen cycle, that the total inventory of fixed nitrogen is sort of constant and steady state over time. I don't really have a lot to contribute to that argument. I'll just say that if it were out of balance, we would very shortly have a lot more, way too much or way too little, and we would certainly notice that. So the nitrogen cycle of the ocean, the nitrogen inventory of the ocean includes both land and ocean, and the main thing that happens in the nitrogen cycle is that it cycles. There's a lot of internal cycling in both the ocean and on land, and so the numbers in these internal cycles are really huge compared to the numbers that represent the processes by which new fixed nitrogen is added to the system in terrestrial and marine environment, and by which it is lost, so denitrification. And that is a presumably nearly steady state pristine or natural nitrogen cycle as far as we can tell before humankind added the red arrows, mainly by adding new inputs through fertilizers and fossil fuels, and by enhanced agricultural techniques to encourage the nitrogen fixation by bacteria associated with plants. And this most of this new nitrogen is added directly to land, but of course that leads to a bit of extra runoff, and so you have an increasing nitrogen input into the ocean. And again, even though these numbers are small compared to internal cycling, the human impact has been to essentially to double the input of fixed nitrogen into the ocean. And so I've always wondered if the marine denitrifying bacteria are ready to step up and keep the nitrogen cycle in balance, right? So if we keep adding, if we double these kind of numbers, then eventually that will have a big effect on the ocean. So there has to be some, the denitrification presumably is stimulated to take care of that. We'll see if that, what controls that is part of our question. So the reason that we're interested, the too little aspect of nitrogen is that it limits production on land. And so that's why it's so important and historically was so crucial to develop fertilizers and increase the production of plants and food for people. And by the same token, if nitrogen is what limits productivity in the ocean, then adding more will add more productivity to the ocean. And that could be a little is good, a lot more is bad. And is evidence, of course, that nitrogen is a really important limiting agent in the ocean. This is a map, two maps that show the concentration of nitrate in surface waters at the time of the yearly maximum and at the yearly minimum. And in both cases, there's almost no nitrate in most surface waters of the ocean, that being the main form of nitrogen that phytoplankton utilize. That implies that adding more would increase primary productivity. A little bit more sophisticated way to look at limitation is this model, and I know that this has been updated, but it shows that for both small and large phytoplankton, vast areas of the surface ocean really are nitrogen limited, and so if you add more, you would expect to find more productivity. And if you look at the way that that plays out in the ocean, this is a vertical scenario of the nitrogen cycle. So the net inputs are the nitrogen fixation, which generally occurs in the surface ocean by photosynthetic microbes. This is the big internal cycling pool, and the net outputs are anammox and denitrification. And those only occur, both of those metabolic processes only occur in the absence of oxygen. So nitrogen loss is anaerobic. And the main places where that occurs in the ocean are coastal or hemipelagic sediments, shallow sediments around the edge of the ocean, and in a few discrete areas called oxygen minimum zones, which only occur in 3 different places around the world ocean. And so they're defined by really low oxygen saturation at a sigma T level of 26.5, which is about the center of the oxygen minimum zones. So these are the 3 big places where oxygen is really, really low, almost zero. And those lead to, they allow the microbial processes that occur in the absence of oxygen and create this entity called N star, which is sort of the missing fixed nitrogen. Oh, wow, thank you. And that happens in 3 places around the world ocean in the sort of open ocean, close, not including close to land, the Eastern Tropical North Pacific, the Eastern Tropical South Pacific, and the Arabian Sea. So those are the oxygen minimum zones. And they comprise the volume of water where, now there's something really bright in my eyes. Is that, yeah, sorry, can't see it one way or the other. And so it's in these 3 places where the oxygen is depleted enough that denitrification and anammox can occur, the 2 processes that lead to the loss of fixed nitrogen. If you take a vertical slice through one of those, so I'm going to talk about all 3 of those here and there to make whatever points, so they're very similar systems, it turns out. In the Arabian Sea, in this vertical axis, you can see evidence that nitrogen loss has occurred, that denitrification has happened because there's this big bite out of the nitrate profile, and there's a secondary nitrate maximum. Those are characteristic chemical features of an oxygen minimum zone. And that the process that we've always assumed was responsible for the loss of that nitrogen, nitrite and nitrate, was denitrification, which is just an anaerobic respiratory process in which nitrate is used as the respiratory substrate instead of oxygen. And it's a heterotrophic process generally that uses organic matter and decomposes it, remineralizes it during the respiratory process to release inorganic nitrogen and nitrogen gas, N2. And you can set up various stoichiometric relationships to burn average organic matter and get slightly different outputs. But if you're starting with average marine organic matter of something close to the Redfield ratio, then the amount of inorganic nitrogen and N2 gas that you can get out of that average organic matter is very highly constrained by the Redfield ratio, by the ratio of the initial material. And so in this permutation, we get, it's set up to get equal amounts of ammonium and nitrite, as well as a lot of N2. That ammonium and nitrite can be further turned into denitrified in the non-canonical sense, turned into unfixed nitrogen into nitrogen gas. And so if you compute the amount of N2 that was produced by anammox, it's 29% of the total, and the other 71% comes from denitrification. And that turns out to be a really strong constraint, because in a system where you are not able to truck in nitrogen from elsewhere, then the only source for anammox is from the decomposition of organic matter, and that's the most that you can get out of it. Anammox and denitrification both lead to the loss of fixed nitrogen, but they're really different processes. So denitrification is heterotrophic and is based on the consumption of organic matter. Anammox is autotrophic and fixes CO2, and so all you need is the inorganic nitrogen, and if you can get it from somewhere other than denitrification, then you're not subject to that stoichiometric constraint. It's a really hard way to make a living, too, because the only source of reducing power is this oxidation of nitrite to nitrate, and you fix just a little bit of carbon for turning over a lot of nitrogen. And so this leads to the expectation that these 2 processes occur in this ratio to remove fixed nitrogen from the system. And in these, this work, it was part of Bonnie Chang's thesis when she was at the University of Washington. She did this elegant, simple but elegant approach to show that, yeah, that really is what happens. And so she calculated and measured and calculated the nitrogen deficit by 2 different means. And this basically assumes a starting material of this recipe with an N to P ratio of 16. And in simple terms, you basically calculate how much nitrogen is missing based on how much you would expect to be there on the basis of the Redfield ratio. And that's 2 different ways of doing that in those symbols, the circles and the triangles. And then her major contribution was to actually measure the amount of excess N2 gas above what you would expect to be there based on saturation at the chemical characteristics of that piece of water. And that's a totally independent measurement. So it's just the amount of N2 gas that's been produced by the combination of whatever processes are producing it. And you see that they match really, really well. And she's done this for the Eastern Tropical South Pacific, the Eastern Tropical North Pacific, and the Arabian Sea. And what it means is that by these two independent measures, you find the same amount of total denitrification or total nitrogen gas production has occurred, and it basically says that it's happening by red field stoichiometry. And so that implies that whatever the ratio, whatever the input of organic matter is, it's probably pretty close to red field because that's the amount of N2 gas that you get from it. And so this was not big news. It was a really nice sophisticated measurement because it's hard to measure excess N2 at that level. But it wasn't big news, but it was interesting because up until very, very shortly before this time, the actual rates of the processes of denitrification and anammox had never been directly measured. And that was partly because it was a hard thing to do against that background of a lot of N2 in the water. And that problem was solved by the Danes, who did two important things: utilized tracer experiments to label up the nitrate or the nitrite and measure it in N30, N2. So this is the smoking gun for denitrification. You have to combine two of these to get an N230. And for anammox, the smoking gun is to label up N15 ammonium, and you get N229 from that because it combines with some nitrite in the seawater that's usually 14. Of course, you get N29 from one of these as well, so denitrification produces both, but if you get N230, then you had to have denitrification. So various combinations of tracer experiments are used in this kind of approach, but these are the two critical ones that enable you to pick apart the total N2 flux between the two processes. The other key innovation was to use these cute little tubes called excitaners, in which the key innovation was that you strip them using helium. You remove the initial nitrogen gas background. So even little tiny amounts of N15 labeled N30, N29, N2 can be detected in the helium background. And so using this method, they and many others have adapted it, including us, to measure directly the processes. And by using a tracer, you may not get the absolute in situ rate, but you really show that the label went from the substrate into the product. And so you demonstrate that the process was happening. So the way that this works is that you measure a time course over time of the accumulation of the labeled N2, and these are data from Hammersley et al. In the eastern tropical South Pacific. And it shows the various combinations of tracer experiments that you usually do. And the key thing is that they always found the accumulation of N29, N2 in the dark diamonds, and they never found the accumulation of N230. And so all this time we've been thinking that denitrification was the main loss process, and we understand from stoichiometry that it has to be the main loss process, and yet when they actually measured it, they didn't detect any denitrification. And they weren't the first people to do this. It was done a couple of times in the eastern tropical South Pacific. And these are data from Tamdrup et al. And slightly earlier visit to the same place or similar location in the eastern tropical South Pacific, and these are the rates of anammox in 3 stations at a couple of different depths measured with that same suite of tracer experiments, and they only found anammox as well. And so this led Phyllis Lamb et al. To suggest a nitrogen cycle that did not even need denitrification. And so you can supply the substrates for anammox by by mineralization, and it was proposed that some of that was actually microaerophilic, so utilizing oxygen in the upper part of the oxygen minimum zone, and also by ammonium oxidation, so that would be aerobic ammonium oxidation producing the nitrite. And in this model, most of the nitrite that was necessary to support anammox actually came from aerobic ammonium oxidation. And you can see how this might work in the upper edge of the oxygen minimum zone where there might be enough oxygen to support low rates of nitrification. But to my mind, there was a critical step missing, which is there was no nitrous oxide in this nitrogen cycle. And I haven't shown you any profiles of nitrous oxide yet, I will, but it's a characteristic feature of the oxygen minimum zone. And so if we're going to explain the nitrogen chemistry of the oxygen minimum zone, we kind of have to have a process that produces and consumes nitrous oxide, and that is denitrification, because anammox really doesn't do that. And so the dilemma, this left us with a bunch of questions. How can anammox dominate the nitrogen loss process if it depends on denitrification for the substrate? And in the open ocean oxygen minimum zone, you're away from the coast, there's no autochthonous source of the ammonium and nitrite that's required, then that is a real dilemma. And so one of the solutions that we suggested was that it had to be a matter of spatial and temporal variability. Those data that I showed you and all the data to that point that showed mainly anammox in the ETSP were really only from a couple of visits there. And so it wasn't like we knew it was a global phenomenon, so maybe we just missed it. Maybe it happened over here and not at that time. And what could possibly account for temporally and spatially variable denitrification? And the obvious answer was organic matter limitation. And we had shown in a longer-term incubation experiments that both the ETSP and the ETNP were carbon limited. If you add more carbon, you get more denitrification. And so our idea was that organic matter supply was what limited denitrification and ensured that it was episodic. The other thing that we suggested that one should consider, of course, is that the community composition might be different. It was conceivable, although not likely, that denitrifiers just didn't like the eastern tropical South Pacific. We didn't think that was very likely, and that wasn't the answer, so I'm not going to tell you about that. We did then approach the problem using the same tracer approaches that others had, and also added our new smoking gun, which was the use of nitrous oxide doubly labeled N46, N2O, and that also yields N230. And since anammox doesn't really do much with nitrous oxide, that's another clear signal of denitrification. And these are Sylvia Newell's data from the Arabian Sea from 2007. 3 stations, 3 to 4 depths per, with all the different possible tracer combinations that I showed you before and more. I just want to draw your attention to the black bars and the gray bars under them. Those are the 2 typical characteristic measurements for denitrification. So that's the amount of denitrification that you get that you calculate after adding nitrite labeled with N15, and that's from nitrous oxide. And for most of the 11 depths here, we detected denitrification at all 11 depths, and at most of them, the rate that you calculated from these 2 tracers were not significantly different. So they both indicate the rate of denitrification. At those same depths, we actually did also detect anammox at 4 of the 11 depths, generally at lower rates, detected using both tracers, although not very reproducibly from the nitrite. So this was interesting because in the ETSP it had only detected anammox, no denitrification. In the Arabian Sea we detected mainly denitrification, but also anammox. Then the ratio was 90% in that range of being denitrification. So that was all done with the excitators, which are very convenient. You can do lots of replication, and they're really small and easy to handle. The other approach that we used was the big bags, which have their pluses and minuses. The plus is that it's a 8 to 10 liter volume of seawater, so it's much less disturbed, much less perturbed than an excitator. There's no flushing involved. There's no replacing the nitrogen with helium, so we rely on Bonnie and Al Duval to have the sensitivity to measure the accumulation of a very tiny signal of labeled N2 in the huge saturated background of N2 gas. And Bonnie published these data recently, just measuring the denitrification and anammox in those bags. So this is in the eastern tropical South Pacific, and 2 visits to the Arabian Sea. The black bars are denitrification, and you can see that in the bags, after about a 2-day incubation, which is on the order of the same length of time that we did in the excitators. This is a single endpoint measurement, however. Denitrification was always detected and usually the dominant thing. So that didn't really help us figure out what controlled why we did observe differences between regions. So in the Arabian Sea it appeared to be mostly denitrification, and why we didn't detect that in the eastern tropical South Pacific. But this has been my working hypothesis for years, that they both occur at significant rates in the OMZs, and it's a matter of spatial and temporal variability. So denitrification, we suggest, is episodic and driven directly by the organic matter input. But overall, if you measure it enough times and places, you'll find that denitrification and anammox occur in a ratio of 71 to 29, or something very close to that, because it is totally constrained by the organic matter recipe. So if denitrification is episodic, the other side of that is that anammox must be kind of slow and steady, like the tortoise and the hare kind of thing. And so we looked again at all of those data from the big bags and the excitators, and all of the denitrification is in blue and anammox in red at several of our stations. These are all combining all of the stations from the Arabian Sea and the eastern tropical South Pacific. And it's not a whole lot of data, but yes, there's more variability in denitrification, and we did not detect it at every station, so it's more spatially and temporally variable, and when you find it, it's usually faster than anammox. The Dalsgaard et al. Had a better data set from a coastal transect along the eastern tropical South Pacific, in which they essentially showed the same thing. So they had a larger data set. It was easier to see the distribution. This is all anammox. At all their different stations, it was detected at all but one of them. And here's the denitrification at the same stations, and it was not detected at most of them, but when it was faster, and sometimes by a lot faster than anammox. So the top of that box is this red line. So you can see that there's a lot more variability in denitrification, and averaged over all of their stations, denitrification was like 71 to 79% of the total nitrogen loss. And so it feels like that sort of solved the problem, that it indeed was spatial and temporal variability and over time. They also showed that when they looked at this a different way to ask whether denitrification and anammox co-occur, and the answer is they don't. So when you have high denitrification rates, you have really low anammox and vice versa. So they're both anaerobic, they depend on each other. The anammox depends on denitrification at least, but they don't occur, they're not tightly coupled. And so that, again, supports the fact that maybe anammox is slow and steady. Look at the axes, much lower than denitrification, and denitrification much more episodic and variable. They're both constrained to really low oxygen, though, so it's not a matter of occurring under different conditions, although in different spaces and times. The highest rates are always under the lowest oxygen, and you get really low rates under measurable oxygen. And so they're both constrained to the same kinds of conditions. So that left us understanding the sort of overall picture of what controls the contribution of the two, that it has to be that overall general stoichiometry. But why, when you go to a certain time and place, do you measure one or the other? And so we addressed that by directly adding organic matter to the incubations and asking if that stimulated anammox or denitrification or both. And so these are, again, big bag experiments, and it's only denitrification, not anammox, shown here, at two different stations. This is one station, this is another, at two different depths. So the different colored bars are different depths. And this is the control with nothing added except the tracer. When you added particulate organic matter or dissolved organic matter, sure enough, you did stimulate denitrification. This is in the eastern tropical South Pacific. And I want to tell you what the organic matter was, because it was particulate material that was captured by Rick Kyle in his sediment traps at the same place and time that we were doing the incubation. So this is what they would have had for lunch anyway that day. Right? That was the stuff that we captured it and then added it to the incubations. And sure enough, that stimulated denitrification. And that was mainly the only thing that stimulated denitrification. So Andrew Babin addressed this in his thesis work with excitainer experiments. So the previous was the big bags. And these are the average and standard error of 4 different experiments performed at 4 different depths, 2 different stations in the eastern tropical North Pacific. And what we did was add nothing, add sucrose, casamino acids, or particulate organic matter to the excitaners. And the particulate organic matter was, again, the stuff that we would catch in a sediment trap that very day. The sucrose, I'd like to acknowledge the Thompson for that, because when we arrived on station, we thought we need a substrate that doesn't have nitrogen in it. And we didn't have any, so I lifted a packet of sugar from the galley. And so I'd like to acknowledge the Thompson for their contribution to this experiment. It turned out really well. So this is the control amount of denitrification and anammox without adding anything to it. That's what they were doing when we collected them. When you add sucrose, sure enough, you enhance denitrification, but not anammox. The gray bars are what you would have expected, the amount of anammox calculated based on the carbon to nitrogen ratio of the organic matter added. And you can see that the measured amount really matched amazingly well to what we would predict on the basis of stoichiometry. So all of the additions of organic matter enhance the rate of denitrification. Most of them enhance the rate of anammox. This one is a nitrogen-rich substrate, so you got a greater enhancement of anammox. This one was added, these were both on the order of 20 or 30 micromolar organic carbon. This was on the order of 5. So the one that, the organic matter addition that was the lowest absolute concentration of organic matter that was added, they liked that the best. And like I said, I think that's because that's what they were used to eating. That would have been lunch anyway. So that stimulated denitrification the most. Along with these experiments, we also added a little bit of oxygen to the controls just to try to parameterize where within the water column they could occur. And they both really like no oxygen at all, and there was not a significant difference between their oxygen tolerance of the two processes. So that means that if you thought that they might have a different oxygen tolerance, you might see different places in the oxygen gradient at the top of the OMZ where one would be favored over the other, but that apparently is not responsible for it. So if you look at the predicted fraction of anammox, which is the rainbow colors in the background, versus the measured, you see that it's really right on. And this solves the dilemma by showing that this, in combination with the spatial and temporal data, that the absolute rates of denitrification and anammox and the ratio between the two rates is directly dependent upon the amount of organic matter that they have available and the stoichiometry of that organic matter. And so that means that mainly in the open ocean, it's going to be red field. Both occur really at pretty much zero oxygen, and thanks to Niels Peter and his STOX sensor, we know that there really is zero oxygen in the oxygen minimum zones. And so that means that anammox is slow and steady and usually detected, but denitrification does most of the work. And that was a sort of synopsis of that scenario explaining the control of the processes by particulate organic matter. And I really do know that diatoms are part of the phytoplankton, but I didn't draw the picture. But I think it's nice anyway in terms of a summary of the two processes being potentially linked and dependent upon each other and resulting in this sort of bottom line. So back to the nitrogen cycle for the last part of the talk, which revolves around a nitrous oxide. So one of the reasons, of course, that we were interested and concerned to find denitrification in the oxygen minimum zone is that we know that there is a lot of nitrous oxide there, and it shows a really characteristic profile. And there are many reasons to be interested in denitrification. This apparently is what the soil sciences like about it. And this is a cartoon from this guy who does soil science. The allure of denitrification is being able to sample the N2O yourself. But the real reason to be interested in nitrous oxide, of course, is its inexorable increase due to the increase of nitrogen loading in the environment. And this basically comes from agriculture and fossil fuels. And the reason that nitrous oxide is of concern to those to people beyond its cycling in the ocean is that with the most of the success of the Montreal Protocol and its various permutations, the CFC load of the atmosphere and emissions to the atmosphere have been greatly reduced, and that leaves nitrous oxide as the main ozone depleting chemical being released today. And so its importance will continue to increase in the future. It's also a really powerful mole-for-mole global warming potential. And so where it comes from and how it's cycled in the ocean is worth understanding. And for us, we were interested in it mainly because it is one of the suite of nitrogen compounds that shows a really characteristic distribution in the oxygen minimum zone. So these are data that Nick Peng put together. Amal measured the nitrous oxide, and Aaron Morello all the nutrients. So this is in the eastern tropical South Pacific. No, sorry, eastern tropical North Pacific. This is the oxygen strong, deep, thick minimum zone. This is a characteristic ammonium peak, really sharp gradient, 2 nitrite peaks of primary and a secondary maximum, a nitrate deficit here, and a very characteristic nitrous oxide distribution. And so when you see that, you know you have an oxygen minimum zone. And what's interesting, I think, Jim mentioned that I started graduate school in 1976. Thank you very much. It's been decades that we've known that these kinds of distributions existed and characterized oxygen minimum zones, and we know the processes that are involved in producing and consuming these chemicals, but we really don't know what poises them to look like this all the time. And they must be very dynamic processes, because those are really sharp gradients. And Al Duvall put together this picture of some of our data from recent cruises, plotting everything on a neutral density profile to make it easier to see the direct connections between the features. And I'm not going to spend a lot of time on this, but it really implies, along with the depth-adjusted distributions, that these things are connected, and there are lots of biogeochemical transformations that are going on that must be connected and sort of poised somehow to maintain that. And we were really struck with this in the ETNP when we compared these two profiles of nitrous oxide. So the red symbols are Yamagishi et al. They collected this profile in the year 2000. 12 years later, we collected the one in the open blue circles from a station about 70 kilometers distant. And this makes you think that every time you go back there, you're going to see exactly the same thing. But it's not a static feature. Obviously, gradients that sharp would disappear in a really short amount of time thanks to diffusion. So there must be a lot of production and consumption going on. And how they're poisoned, how they maintain this over years is a really interesting question, and we would love to be able to investigate that. So one of the things that we've done was to try to measure directly the production and consumption of nitrous oxide. This is, again, another chapter from Andrew Babin's thesis, in which he used our tracer N2O, N46 N2O, consumption of N2O into nitrogen gas. And I'm not going to show you the profile that I just showed is from one of these stations, so you sort of know what the nitrous oxide profile looks like. And these are his measured consumption rates in the black squares. And using a 1D model to calculate the necessary production, the production that was necessary to maintain the measured concentration profile. We calculated those, and those are the open diamonds that you can't really see in most places. They're here, but they mostly overlap almost exactly on the consumption, implying this is a really tightly coupled system. You have the highest production and consumption where you have the nitrous oxide concentration maximum. And the places that they don't match, where you have actual production in excess of consumption, is near the upper edge of the oxygen minimum zone in the oxycline. And that's where you might have some nitrous oxide accumulation, and that's also the nitrous oxide that might actually reach to the atmosphere. It might be available for emission to the atmosphere. So the turnover times that he calculated, this is the oxygen distribution. The turnover times that he calculated on the basis of that range from a day or so in the concentration maximum, where the rates are greatest, to actually years when you get up into the surface layer of the ocean. And the main consumption term, of course, is denitrification, but denitrification is also an important production term, which is something we might not have really appreciated. And so that implies a very dynamic cycling of nitrous oxide in the oxygen minimum zone, especially near the oxycline. And nitrous oxide is also produced by nitrification in the surface water, and that's another part of the puzzle, the production rates of nitrous oxide. So we were really interested in this paper and talked about it in a lab meeting because it identified surface nitrification as a major uncertainty in determining the magnitude of the rate of nitrous oxide emissions from the ocean. And this was a modeling paper where Zamora and Oshleys investigated the control of nitrification and where it happened, how fast, and what determined its rates, and then the yield of nitrous oxide from nitrification. And the two main uncertainties that they identified, and I think they had 5 or 6 of them, but these were the two big ones. What is the actual size of the nitrification source, so how much nitrous oxide is produced from nitrification, and what is the oxygen concentration at which we switch from aerobic to anaerobic processes and from net production to net consumption. And we like that because we can help you with that. And I hope some of you saw Jimmy G's poster this afternoon in which he addressed this directly by measuring the production of nitrous oxide from various substrates. So again, in this case, not labeling up the nitrous oxide, but labeling the ammonium or the nitrate and nitrite. And presumably, if you get nitrous oxide after you've labeled nitrate and nitrite, it came via a denitrification pathway. If you get it from ammonium, it could come directly from some magic that happens in ammonium oxidation. We're not quite sure what that pathway is. Or it could go all the way around and be coupled nitrification, denitrification, or it could be nitrifier denitrification, but it started out as ammonium. So using this combination, these 3 independent tracer techniques, tracer additions, he measured the production rate of nitrous oxide. And he actually shows the production rates on his poster. But these are preliminary data that just show the actual accumulation of the label in nitrous oxide from the 3 substrates. And you see that ammonium in blue is a source of nitrous oxide in the surface above the oxycline and within the oxycline. So right where oxygen gets right about to 0, you still have a little bit of production of nitrous oxide from ammonium. But the biggest signals are from the reduction of nitrite and nitrate. So, as I said, by coupled denitrification, nitrification, but mainly from denitrification. And you only see that pathway in the heart of the oxygen minimum zone, where presumably, where we know, in fact, that from independent measurements that nitrification is not happening. Ammonium oxidation is not occurring. And so this implies, together with the production and the measured consumption, and the calculations on turnover, that there's a lot of nitrogen transformations going on in that apparently steady state nitrous oxide profile. So we have really rapid turnover of nitrous oxide at low oxygen, primarily in the oxycline and within the oxygen minimum zone, and both nitrification and denitrification contribute to the production and denitrification to the consumption. And so, as I said, this apparently poised homeostasis in the depth profiles of nitrous oxide and all the other chemicals that we observe in the characteristic of the oxygen minimum zone belies what has to be really dynamic biogeochemical processes. And as I said, I think Rachel Carson would have approved of this investigation because nitrogen is one of the things that has an impact on the quality of life and the ocean and the environment. It's similar to the kinds of concerns that she originally raised. And I'd like to acknowledge either Mark Altabet or Nick Bauskell for this picture. They both took it, and I can't remember who sent it to me. So there's a picture of Rachel Carson. There's a statue of Rachel Carson in the yard at the MBL. And she would have been a nitrogen babe. So I'd like to thank the Ward Lab past. These are all the past nitrogen babes. You see that there's a good representation of both genders. I thank them all very much. And thank you.
We do have time for a couple of questions, and so speak up because I can't see.
You can't see a thing from here.
That helps. There's one in the back.
This is a former nitrogen babe.
Hi, Beth. Great talk. So I was thinking about what you were talking about with the sort of decoupling in time of denitrification and anammox, but the fact that they're linked via stoichiometry. And I guess I was wondering why we still don't see accumulation of ammonium, and if we just haven't looked hard enough, or if you think we should find it where there's a lot of denitrification going on but no anammox.
Yeah, I don't know. I don't know the answer. It troubles me too. And we should find it, and we have looked pretty hard. So the depth profile that I showed you that Nick Peng put together, that was a combination, Nick, 24, 28 casts at that station? 28. And there was no ammonium below that maximum. And that was with the fluorometric method. So I don't know.
Yeah, did you have ammonium leaking into the top of the OMZ from that peak? I couldn't quite tell whether there was overlap of...
It's a little bit above there, I think.
Okay.
Yeah. So...
Thanks.
Yep, I don't know.
Is there another question?
Where's the wine?
Lynn, come on up and let's. Lynn Talley is future president in January of the Ocean Science section, and I'm Jim Murray, president. We give Beth Ward this certificate for the Rachel Carson lecture, which he can frame, and let's give her a good hand to acknowledge her receipt of this today.